Erudite Chair, Professor Stephen O'Connor, who is a chair at the Royal Academy of Engineering. Stephen, over to you.
Thank you very much, Brendan. So without further ado, I'd like to introduce Professor Roger Staff, who is the head of imaging physics at NHS Grampian, and he reliably informed us about 15 minutes ago that the first snowflake of winter fell this morning. So Roger is going to speak to a title overview of AI in imaging science, from the peak of Mount Stupid to the Valley of Despair. Over to you, Roger.
Thank you very much for the invitation to speak. This is very much a personal journey. I've been involved in with a breast cancer project over the last 4 or 5 years that's opened my eyes to the challenges that we face. So let me just share the window. Okay, thank you very much to Stephen for asking me to do this. I have accumulated quite a lot of knowledge and skills, and I've never really put it together apart from the IPEM meeting earlier this year and presented it to the community. So I do see this as quite a good opportunity. The flippant statement from the peak of Mount Stupid to the Valley of Despair is essentially a description of our personal journey on our particular project, but I'll cover other things than where it's creeping in and is actually in place, whether you know that or not. Okay, so what is our destination? AI, as I said, you know, it's a medical product. And where we want to be is something safe, efficient, have equality and usability. That's always quite a thing, and I think usability is one of the boundaries to adoption. AI has the potential to solve at least parts of the workforce crisis that's going on, and is likely to get worse over the next few years. So all those standards are essentially risk dependent, and the way I've come to think about how we test AI products is really about risk. It is about, you know, there are a whole multitude of AI products that are out there. If ones are actually making decisions about part of the patient management, quite important decision about it, obviously the risk is high, and you want to minimize that risk. Whereas there are some other AI tools that may well be of less risk. I came across one recently with GE with their new big PET scanners, where they don't do time of flight, and they actually simulate time of flight using an AI product, which I was relatively impressed with. So just as a matter of comparison, and I've done quite a lot of drug development studies over the years, and in terms of testing and getting the product to market and seeing it whether it has a clinical impact. I think the parallels between drug discovery are actually quite pertinent at this moment in time, in as much that what hurdles do we put in front of an AI tool? And again, it's risk-based, and if you've got a drug that doesn't do any harm or is not inferior to those on the market, then its route to market is quite easy. Or whereas if it's got massive implications for the individual's healthcare, then it really needs to be tested in the ecosystem where it's going to be used. So I think people are all familiar with the phases, phase 1, 2, 3, and pharmacovigilance phase 4, as it's sometimes referred to. Perhaps we can split up those steps in AI, and there are certain guidelines written by a multiple of groups about how things should be done at various stages. And the thing where we were not actually very mature is producing the safety and efficacy guidelines, which would have been equivalent, in my view, to phase 3. And there's a similar pattern of stages when surgical innovation, where you choose to do it
Roger, you're on mute.
How about now?
That's better. Thanks, Roger.
Thank
Okay.
you.
Would you like me to restart?
If you go back about 30 seconds, please.
Okay. Okay. Right. So the destination, where we need to be, safety, efficacy, equality, and usability. And I've come to think of these as risk dependent and to approach the problem in terms of risk. What risk does any AI tool present to the healthcare pathway? You know, there are multiple AI tools out there, some of them which carry very little risk and some of them carry multiple risks and obviously are key decision makers in the patient pathway. I think it's pretty good for us to run the parallels with other medical developments, development being probably the most commonplace here. I think we're all familiar with phases 1, 2, and 3 of a drug development process and relatively clear about what their steps actually are. Developing a AI tool probably has a little bit more complicated steps, but still we can split them up into phases. You know, can you get it working on your local population? Are you training your data set on the appropriate training cohorts, etc.? Can you roll it out into a separate population? And then finally, can you actually use it in the ecosystem that it's meant for? And similarly in surgery, if you're going to innovate in surgery, then there are various steps, the ideal step that actually allow you to get your surgical technique into clinical practice. AI has multiple problems in as much that doing a randomized control trial is sometimes difficult. What the KPIs actually are difficult to define in the context of the screening, which I'll pick up on later on. Sometimes you're dealing with hundreds of thousands of images that you may not be able to do a randomized control trial, at least at the patient level. You may be able to do it at, say, the site level, which is something we've been discussing recently. Okay, so there are guidelines out there, the Spirit and Consult and Tripod and Charms, and they've all been from various organisations and they all do slightly different things in terms of the evolution and qualification of any AI tool. Everybody's had a go. There's various organizations, NEMA have had to put something forward, etc. As I said previously, the randomized control stage, the last bit of any qualification, and the traditional the traditional badge of a medical product would be a CE mark, but unless it's been tested in the actual ecosystem it's intended to working at, I think the CE mark has limited value in terms of it's an early development badge that you should and see it as such. It's got a CE mark, therefore they can sell it. It's not really an appropriate attitude to take. ISO, as I mentioned, IEEE have had a go. BSI, I think, are probably coming out with some standards in the near future about how we can go about. These will largely be generic, and they can't be anything else but generic standards. So the whole range of how you would test and get something to market is entirely dependent on the AI tool and the risk that it presents to patients. So the flippant statement comes from the Dunning-Kruger effect, which essentially is that the more you know, the less you think you know, etc. I've been at the peak of Mount Stupid, having been relatively familiar with drug development rather than testing of AI tools. And then I fell off a cliff. I thought, how on earth are we going to prove this? 3 or 4 years ago, how are we ever going to prove that this works in situ, as I say, in the ecosystem that we're working? I'm pleased to say I think I'm edging my way up the slope of enlightenment, but we'll wait and see. Okay, so there are things out there at the moment, and I'll just pick a few. This is by a company called Subtle MR, which allows you to acquire MR images quickly, and it's an AI tool that resamples in case space using AI inferences. By and large, if you're going to say quality is the task of identifying the high signal in one of the vertebrates, then these two images are effectively equivalent. It's at the boundaries where you start to see things differently, and it falls off at where that boundary actually is, and the operational gains that the AI tool actually gives you is a balance for your operational needs. I've been quite impressed with all of the speeding up MR tools recently, to the extent that there are conversations with the Scottish government about actually rolling this out as a single project. As you can see, it generates additional resources and additional time slots that cuts down a lot of MR time very quickly, and is a relatively cheap investment. So the next stage, again, is from the company called Subtle, is looking at accelerated PET, so that there's a whole body 24 image on the left and one that's acquired in 6 minutes. And again, the images, just in terms of perception, are different. Whether they are clinically inferior is something we need to convince our radiology friends and of. But you can see how it improves, well, or rather, it produces an image that's probably of equal clinical value. PET time, as we know, is one of the burdens currently in service delivery, and this allows you to put through images very quickly. Now, this is coinciding with the PET revolution of large field of view images, or rather PET scanners, where we've gone from typically 11 and 12 centimeter fields of view to 32 centimeter fields of view, and whether this advantage, the subtle PET, is maintained with the evolution of the upstream technology, we'll have to wait and see. So just talking about subtle MR, about the dimensions that we've started to look at. So in terms of safety, we can also always measure the quantitative measures, noise and contrast, etc., and it seems to be reasonable. As I say, the images are different. Efficacy, does it give us any operational gains? Yes, it does. There's been no obvious change in accuracy. Equality, none has been there's no expected inequalities from what we're doing, unlike some of the other tools that I'm going to talk about. Usability, yes, it's automated into the workflow. Nobody sees it. The subtle tool sits between the actual acquisition and the pack station, so that's where it's actually intervened, so nobody sees there's no messing around with sliders, etc. So the risk, the provenance of the vendor produced or commissioned, we're in the early days in terms of provenance. Can you trust this third-party company? Although GE and Siemens, they do provide additional speed-up measures. So if you're happy with the Siemens machine, etc. So the risks are, do these quantitative measures actually translate into health gains? So all of the physics-like measures that we've done, are they actually translating? And the numbers are pretty small at the moment in terms of number of sites, and in order to do this properly and see whether we've missed something or something is detected that's not there, etc., we're going to have to wait and see. And really, in terms of this product, we're in that phase 4 type of environment, and there may well be unexpected inequalities. It may not work for morbidly obese patients, etc. So this is MVision. It's a tool to segment up patients into various marks, particularly in the neck. It works quite well in the neck, in order for you to identify dosing, etc. From CT images. As it also works with MR, again to assess how we're going to. So the comparison that's been done is done with human observers. So by and large, the regions of interest are almost identical, but the variance between a human observer and MVision is the same as the variance between two observers drawing the same regions of interest. It increases operational throughput, and that's part of the way that they've tested in as much that people can do more in less time, and that's good. There was no change in accuracy as I did. Yeah, and what it did do was improve consistency. Now, within this model, you're allowed to adjust those regions if you think you've got it wrong, etc. There's no expected inequalities. Again, the numbers are small. You're in the phase 4 of this type of product development. It's easily automated in the workflow. Again, the risks are the provenance of the vendor. This is a third-party tool. I've not come across any GE and Siemens equivalents to this, but I'm sure that they're in the pipeline. Again, the risks are, do the quantitative measures translate into health gains, and if there are any unexpected inequalities, such as people with clips, etc. List X is essentially about fixing the orientation of an ultrasound. So when you put the orientation on, it continue, and as you're aware in say cardiology, you need to record fixed planes of interest. This works really well. It allows, it's easily tested comparison with human expert observers, expert users. Again, as with most AI tools, the key selling point is about fixed, putting more people through. There are no unexpected inequalities. It's easily worked into the workflow. It essentially appears as a slider on the tool, etc. And it goes green when you've got the right orientation. Again, third-party vendor. The quality measures may or may not translate into operational gains if you bring it on board locally, and whether there's any unexpected inequalities. Right, so now onto the main part, and we've been doing this for 4 years as part of the ICARE funding stream. MIA is a tool that was designed to screen breast cancer screening. So to say yes or no to recall or not recall. That's not yes or no to cancers, but to recall or not recall. This is how we tested it. We started off with 93,000 screens, and we filtered various screens out. It doesn't work for everybody, so this is a real world retrospective sample that we have at least 3 years follow up on. Okay, so the project rulebook. We started this prior to all of the guidelines. So by and large, our project rulebook translates onto the guidelines pretty well. No interaction with the vendors, so we're going to test it away from them using real-world samples, abide by data governance, etc. Focus on operational gains, which is where it's going to sell, we're using the dual reader process as the gold standard. So humans are the gold standard here. There was a snakes and ladders game to get approval. It was a huge amount of data, the PBPP, and this is one of the obstacles to the implementation of AI, is actual permissions to test it on large NHS data sets. So we did all this in a safe haven environment, which included us being able to process the images in a safe haven environment. That was produced by Canon. So this was a multi-collaborative project. Okay, so the data was trained on about 145,000 Middle European breast screened patients, and it produced an ROC curve. So what you need to do here, we first tried to use it out the box, and we had this operating point. So above a certain value, we were going to recall them, and below a certain value, we weren't going to recall them. And as you can see, it didn't do very well. It was actually quite sensitive. We did get lots of recalls, but it wasn't very specific, which meant that we were recalling almost half of our patients if we had have implemented this tool. The cancer detection rate essentially remained the same. This then led us to the conclusion that we needed some local optimization, and we've shifted the point from the out the box solution to one that was recommended by the vendor to maximize our local production, and that's essentially moving the X on the ROC curve. And it improved, life improved. So we've got with our updated threshold, we're now down to 12% recall. We never really had a significant impact on our cancer detection rate. Now, just for comparison, a single user using those 45,000 images will produce these types of figures. So a single user will be more specific but less sensitive. But then again, it will recall almost half of the patients. So if by using this tool, we're saving human reads as being, we are paying a downstream cost of increasing our recall rate, which means there's more ultrasounds. There may be consequently more fine needle biopsies or MRs or additional. So a full health economic assessment of this tool will probably come out cost neutral. I wouldn't say that it would increase our costs, which again defeats the object. So we were disappointed. This is our standard double reading system that occurs in most. We took a deeper dive into the data and looked at our recall rate over time. And what we saw was you get these normal recall rates, and then suddenly there was this step change in the recall rate. That step change coincided with the software update of the original system. So it was an upstream change that the AI tool was not allowed to did not cope with, which was a surprise to them and a surprise to us, which means your tool has to be constantly retuned to the local data set each time something is changed. Not helpful. So the take-homes tool was not an out-the-box solution. Local optimization is required, and that optimization may well be redundant once you've had upstream changes. Okay, again, our box is safety. Efficacy not proven. There was a query about race. We didn't really have enough diversity in our sample, but those of African origin tend to have more denser breasts, and the performance looks as though it was less. So it's not, it's not robust in that sense, and there was clearly an inequality going on there. It was useful, usable, it was integrated into workflow. The robustness was an issue. Whether it's acceptable to patients and stakeholders is the next bit I'm going to cover very quickly. We asked the patients whether they approved of it, and generally the answer was yes. And we asked radiologists whether they approved of it, and the answer again was yes. But what they wanted, what evidence they required, did actually vary. This is for the patients. The patients were generally up for it. Moving forward, we're introducing MIA as a blind reader. This is the strength of in a prospective study. You would have to test this prospectively, and MIA does have an advantage of recalling more patients with cancer. That's cancers that have occurred, that either were missed at the time of screening or occur between two screening incidences. So there's probably cancer there. So we are picking up in the prospective study more cancers, but we are recalling for additional reads as well. So what the balance there is essentially watch this space. So the prospective study is essentially what we're doing now. There's 15,000 of them, 3 months worth. There's no software upgrades on the machine, so we're fixing that. So it's not quite in the ecosystem that we were thinking about. And our outcome measures are cancer detection rate, recall rate, and time to reporting. What we found is that when they had this, the MIA outcome, and it draws regions of interest on a mammogram and say, look at this area a little bit closely, etc. It did increase throughput. So I'll stop there. And apologies for the breakdown in communications earlier on.
Thank you very much, Roger. Very interesting indeed. We look forward to seeing further results. If people have questions, please, could you put them into the chat and we'll deal with all the questions at the end, and we can move on then. I'd like to introduce Ashley Ballard. Ashley is an advanced practitioner and digital pathology lead at the University Hospitals Dorset NHS Foundation Trust and the University of Portsmouth. Ashley is going to speak to a title, Enhancing Histopathology with AI, Opportunities and Challenges. Ashley, the floor is yours.
Okay, so thanks for that introduction. Just see if I can share my screen, bear with me. Okay, so hopefully that is coming up for everyone. So my background is cellular pathology, histology. Now I think unlike maybe some of the other disciplines in this presentation, histology AI is still very much a work in progress. There are some early adopters, but most departments in the UK are still getting systems in place. So this is very much looking at where things will hopefully be in the future, rather than necessarily where things are right now. Now, I think for histology, it's very much about productivity. So one of the things I wanted to look at was the issues facing cellular pathology and why we need to consider artificial intelligence as a strategy to resolve them. Look at some of the aspects of AI with relation to cell path and how it might work, where we are in the implementation process, and then some use cases, some things that we'll hopefully be getting in place in the future. So cellular pathology probably isn't alone in facing significant workload challenges, but they are getting quite acute. So data is indicating that we're seeing workloads increasing by about 4% every year, and this was before COVID and all the waiting lists to catch up with those backlogs. At the same time, pathologists are leaving the profession and they get to retirement age, and there's not enough people coming in to replace them. So we're seeing those staff numbers reduced by about 5% a year. Data from laboratories is also showing similar pressures really for the lab staff. Again, workloads increasing and staff levels reducing. All of this means we're getting significant backlogs in the laboratory, turnaround times getting worse and worse, and we really need something to boost productivity levels. And just on the graphic on the right there, you can see there's that growing gap between workload in and people available to complete that work. There are national strategies that have been put forward to try and alleviate this. Three of the key ones really, it's about training some of the scientific staff to take on roles traditionally done by pathologists in terms of dissection and reporting. There's a drive to develop pathology networks in England. So 29 networks have been identified and they're at various stages of development. Although it's not entirely clear really how beneficial they will be for cellular pathology, given the cell path is such a manual process. Networks, I think that's the jury's still out on those really. But in terms of today's discussion, it's all about digital pathology and AI and seeing what we can do with that. As I mentioned, this is still very much a work in progress. We do have funding to try and get this moved forward, but many departments are still going through the process of procuring systems and setting them up. Okay, so very quickly, I wanted to go over what artificial intelligence is and what it means. It's a term that's often bandied about, but what really is it? Artificial intelligence itself is something of an umbrella term. AI is just about automating behavior or making computers that mimic human behavior. A subset within that is machine learning, and this is programming systems to learn. And then another subset, a subset of machine learning, is the neural networks or the deep neural networks, and these are better able to learn features within the data. So basic artificial intelligence, this does have a role. It can be programmed to carry out certain activities, but it's very much dependent on the programming of the system. You have expert systems in place, and these can seem very smart, but it's all down to how much information is put into them. Data is programmed into a knowledge base, and then you have rule-based engines that interpret that knowledge base. These can be used for clinical decision support, but learning with them is limited, really. Image analysis, you have basic threshold image analysis. You define a certain threshold, that is a cut point, and that separates out your groupings. Machine learning, I think the simplest description really I heard for machine learning is you take a group of data points and you draw a line to try and separate them out into groups. And that is really all it is about. So there are these various statistical tools you can use, varying from regressions, poor vector machines, so on and so forth. But these are just statistical tool sets that draw that line. It's all about separating that data out, grouping it into clusters, and it's how you define those cut points that's the key thing. With most of the basic machine learning tools, you do need to define the features. You'll need an expert that looks at the images, marks the images. This is a nuclei. This is a tumor cell. This is a normal cell. This can be a bit of a labour-intensive process. So this can act as a bit of a delay on development. But with machine learning, you can train these systems. They can learn. So deep neural networks, these are the systems really that have enabled the significant developments in AI for image analysis in histology. So at a very basic level, a deep neural network is just a network with more than one hidden layer. So you'll have your input layer, your output layer, and multiple layers in between. These can be trained with supervised or unsupervised learning. And crucially, these are very good at identifying features within the data set rather than individual items. So a feature could be an area of stroma, an area of tumor, rather than maybe necessarily individual cells. The kind of latest, greatest thing really with this is the convolutional neural networks. These are particularly good at recognizing patterns and images in data. The pooling layers within these networks, they're very good at eliminating noise within the data. And because of this, they can be very good with unsupervised learning techniques, which obviously helps if you're short on time and you want to develop these systems. Rather than having to mark everything up, you can just throw loads of data at these systems and they will learn from that data. They're also very, very good at complex features. So by clustering loads of these pooling layers and convolutional layers, they can learn very well and factor in all these very complex data sets. And so one of the issues with artificial intelligence in histology, we still need to produce the graph slides. So in order to apply these techniques, we need to produce the slides as we would normally, then digitize these slides. And this is where many departments currently are in the UK, where we're still in this process of setting up these digital pathology systems. You need scanners that can build up these high resolution images. Just on the right there, you can see how these images are actually produced. Take loads and loads of images of the tissue on the slide, stitch those together as a technique called tiling. This does produce very, very large files, typically around 1 GB per slide. And this does present a bit of an issue for us, really, because obviously storage isn't cheap. We've got to store quite a lot of data. Current guidance, we've got to store 8 years worth of images. So this is many petabytes of data we've got to store. And trying to fund these solutions is a bit of a challenge. So, as I mentioned earlier, this is still very much in progress for many departments. Most departments now, at the moment, they're still in the emerging phase. So they're starting to get the whole slide imaging systems in place. They're starting to scan a few cases, maybe a little use of artificial intelligence, very much testing the solutions, really, seeing what works. The hope is that by 24, 25, we'll be at the maturing phase. So we'll be scanning 50% or more of our cases and using image analysis routinely. Whether or not we'll get there is debatable. I think that there have been a lot of delays along the way getting the funding in place. Okay, so now I just wanted to have a look at some of the diagnostic artificial intelligence use cases. These come in 3 flavors, really. So you have the more basic algorithms that just count cell types. The more advanced algorithms that identify features in those tissues and give more information to the user about what is happening. And then there's some more items that are still in development, but offer some significant promise for service delivery. So this is an example of some counting algorithms. This isn't an exhaustive list, but it just gives you an idea, hopefully, of some of the systems that are available and how they work. So these particular solutions are from a company called Visiopharm, and these are used for giving semi-quantitative scores on some of the markers we currently routinely employ with breast cancer cases. So you've got estrogen receptor, progesterone receptor, HER2 receptors, and Ki67, which is a marker that we use to identify the proliferation index within tumour tissue. So from the images on the right, you can see various cells are being highlighted. You've got the red cells that are strongly positive. The orange ones are more weakly positive, and then you've got the yellows, which are very weakly stained, and the blues that they're negative nuclei. With these algorithms at the moment, the user still has to actually outline the areas of tumour on the slides. So that is potentially an area of error, but I think where these can help, makes things more predictable, more repeatable, and also it saves time. Counting these cells manually, it's a bit of a labour-intensive process. If you've got an automated system that can do all that for you, it is going to be a benefit. The HER2 works slightly differently. This is a membrane stain. So this is looking at continuous connected membranes within the positive tumour tissue. Next, a feature identification algorithm. Again, this is looking at breast tissue. Something to flag up. At the moment, we're primarily focusing on approved algorithms for clinical use, and I'll cover the reasons for that a little bit more later on. So these apps, they do a number of things. They generally come as a suite of tools. So this particular one is from a company called IBEX, and by using these, you're able to grade malignant and pre-malignant lesions, identify micrometastases and metastases in lymph nodes associated with the breast tissue. All of these would still require pathologist review. So with all of these algorithms, it's not about replacing pathologists. It's not about getting rid of those stuff. It's just about providing extra tools, extra ways of providing information that will hopefully improve productivity, reduce error rates, and boost those turnaround times. However, having said that, the sensitivity and specificity of these solutions is very, very good. It's generally close to 100%. Although we're not seeking to replace pathologists, they often do perform better than an average general pathologist. So yeah, the images on the right, you can see how these would be displayed to the user. So you get heat maps on the tissue, highlighting these areas of interest. Another tool we have available is for prostate cancer. So prostate cancer, particularly with screening cases, we tend to use a system now called template mapping biopsies. So multiple biopsies will be taken from the prostate, multiple core biopsies. This is very good for the patient because it provides excellent information on exactly how much the cancer has grown, how much it's invaded. But from a department perspective, that does produce a great many slides that all need to be reviewed. With the shortage of pathologists, this is, you know, if you're spending several hours looking at a single case, it's a delay, and if there's anything that can help alleviate that, then it's obviously beneficial for reducing those turnaround times. And this is what this tool is for, really. Similar to the breast tools, it identifies those areas of interest with heat maps. It provides a Gleason grading, which is part of the diagnostic criteria, and helps with identifying perineural invasion. Again, very, very sensitive and specific. and it does help reduce those false negative diagnoses. Okay, so with this slide here, I just wanted to look at some of the developments, so some of the things that are hopefully coming in the next few years. One of these is looking at ALK and ROS1 fusions in non-small cell lung cancer. Now, this is traditionally done with a molecular test. In England at present, we have a small number of specialist centres that carry out all this testing. So most laboratories, they would send these tests away for molecular analysis. This can take several days to get a result back, sometimes a couple of weeks. If you've got an artificial intelligence tool that provides this data, you can run that tool and potentially get an answer back within a few minutes, maybe a couple of hours. So that is a huge potential gain. Reduces turnaround times, improves patient care. This is still in a research phase, but this is looking at the H&E slides and picking out features. So I think this goes back to what I was talking about earlier, really, with these convolutional neural networks and the benefits that they've had developing these image analysis tool sets. They've allowed these large data sets to be fed through the system, and these tools they identify features within the H&E stained sections that might not necessarily be evident to a pathologist, even a very experienced pathologist. They're very subtle features, and I think this is potentially something that could be rolled out to other test types, and this could be a major benefit in the future. So because we still have to produce GRAS slides and scan those GRAS slides to get to apply the artificial intelligence systems, obviously image quality is critical. I just wanted to kind of go over some of the issues we do face sometimes on this slide here. So in image A, it's a little bit difficult to make out, which is kind of the point, really. On this particular slide, there is actually a blob of wax over the tissue. You might just about be able to make out with this an immunohistochemistry stained section underneath that blob of wax. If this was being looked at on the GRAS slide, the user could just wipe off the wax, look at the slide, no problem. But because this is being scanned, the blob of wax obscures the image and it's no good. In image B, this is kind of more of a scanning issue, really. The scanners that we use, they don't quite scan the entire slide. There'll be a couple of millimeters around the edges, top and bottom, that won't be captured. So if you've got a particularly large piece of tissue that is close to the edge, it'll just cut off that piece of that tissue and you won't see the full image. So this is something we have to consider when we're processing these images, these tissue sections in the laboratory. And finally, in image C, it's a little bit tricky to make out, but if you look at the area of tissue in the green box, so this is out of focus. So this is perhaps where you've got two pieces of tissue on the slide. One is slightly thicker than the other, and because of how the scanner captures that slide, because it only takes that single image through the tissue, one piece is out of focus, and that will be useless for any AI algorithm. So the barriers to implementation. So as I mentioned earlier, in order to apply the AI, first of all, we have to get digital pathology in place. This is a costly process. You need to get scanners in place, you need to get the software in place. There's licenses that need to be paid for annually for that software. And there's the huge storage costs. And this is For an average department, you're looking at several million to implement, and then ongoing revenue costs in the hundreds of thousands. We do have funding from central government to get this in place now, but it is still a challenge to get all this in place. You then need to get that all interfaced with your other computer systems in the departments, your laboratory information system, so you can get that patient data going back and forth. This can sometimes be a little bit challenging, and laboratories in the UK are primarily UCAS accredited. So we go through an accreditation process with the ISO 15189 standards. Every test we do, we have to get that test accredited. Part of that accreditation process, they like to see the tests are either CE-IVD or UKCA approved. Some of the AI applications are, but some of them aren't at the moment. So we have to be careful which ones we select, really. If we can get this in place, the benefits should be significant. Digital pathology alone should boost productivity by 10 to 15%. There's very limited data on artificial intelligence at the moment, but for where it is applied to those tissue types, we could see some really significant boosts, and some have speculated it could boost productivity by up to 40%. But I think that the main benefits really will come with those reductions in interpretive diagnostic errors, improvements in turnaround times, service quality, and patient safety. However, having said that, a lot of these benefits are focused just on reporting workflow at the moment. The benefits for the laboratory, they're not quite so clear at the moment. It could actually reduce productivity in the laboratory because of that extra stage we have to go through. We have to do our existing processes, creating those glass slides, and then we have to do the digital pathology on top of that. So just to kind of make that a little bit clearer, really, I just want to show you this simplified process map of an average UK histology department. So you can see the areas in blue here are the bits that will be done by the laboratory, and then in green you've got that reporting process. At the moment, the digital pathology solutions, the artificial intelligence is very much focused on that that reporting process. The laboratory processes, a lot of them are very manual, and there's not a lot of work that's being done at the moment in terms of boosting productivity in that particular area. This is something that does need work, I think, and that there could be some major benefits here. I think these are just some areas that could be looked at, I think. So case triaging. So if we can triage cases, we can identify those cases that are suspicious, then those can be prioritized. Now, I'm not sure how many of you on this call are familiar with histology, but generally speaking, we're looking at turnaround times in kind of 7 to 10 days. That's our targets. So if we can prioritize those cases, identify which ones need to be looked at first, bring that forward, do those in 2 or 3 days, that's obviously going to be a significant bonus. Automated slide and block quality control. So I mentioned earlier about the image quality and the need to get high quality images for applying the AI. But there are also some other benefits there as well. If we can reduce the number of recuts we need to do, then that's going to be less work going through the laboratory. That's going to boost those boost that productivity, boost those turnaround times there. Currently, IQC is a very manual process in the laboratory, or you might have 2-3 members of staff working full time just checking the blocks against the slides. If you could automate that process, again, that would be a major boost. And reflex testing as well. Generally speaking, the workflow at the moment, cases will go through to a pathologist who will look at the H&E stain sections. They'll then decide if they want to do additional staining to identify certain features. You can have a system in place there that looks at those slides before they go to the pathologist and alerted the laboratory that these tests may be required. They can be done straight away, maybe cutting a couple of days out of the process. Okay, and yeah, so that was all I wanted to go through really today.
Lovely. Thank you very much, Ashley, for that presentation. Again, we'll take questions at the end, and if you could put your questions into the chat, we can line them up. So I'd like to move on to our third and final speaker, Rachel Smith, who is Embryology Innovation and AI Lead for the Fertility Group. So welcome, Rachel. The floor is yours.
Hi, thank you. I'll just share my slides a minute. So, thank you for the lovely introduction. My name is Rachel Smith. I work at Fertility, and as my title says, I'm Embryology Innovation and AI Lead. For those who don't know CARE, we are a fertility provider. We have been around for over 25 years. We have 15 clinics within the UK. We have 3 in Spain and 1 in the US. I'm currently speaking to you from our Spanish clinic, so lucky me. So just to give you an understanding of what we are doing in terms of AI in our fertility industry. So there has been a massive explosion of fertility, and this is down to 2 real reasons. We have been building big data sets of patient records and image files, and obviously there's been a lot of talk about how we use AI, but this has been made possible by increases in computing power. And in terms of an algorithm that we derive using AI, it's there to solve a problem. And as we've already discussed, this is very useful when we're looking at images. And within our field, we generate ultrasound images at follicular assessment for a patient on a patient's ovaries. And in terms of uterine receptivity for an embryo, we look again with ultrasound. And then we have images generated for embryos through time-lapse imaging and through static camera observations. So how can all of this information help our patients to become pregnant and have a live birth? Well, this can be applied indirectly through tailoring of our procedures and our treatment protocols, or directly by looking at the gametes, the eggs and sperm, and the embryos to help identify indicators of quality and select those embryos with the greatest chance of outcome. So we've seen AI in other areas, but how can it benefit in fertility? So we are using it currently as a support tool, or that is the role of AI currently being developed to help enhance our laboratory procedures by saving time. AI can produce results so rapidly and consistently without subjectivity. It can interpret lots of complex data that may be difficult to handle in-house generally. And because it has that potential to save time and streamline practices, it would hopefully save some costs. And because of its implementation doesn't involve very expensive equipment, there is the potential to obviously do this without too much expense. So the areas that we're hoping to look at today will be outcome prediction for our patients, understanding their chances of success, then looking at how we can enhance our treatment decisions through that process when they attend a clinic, specifically looking at gamete and embryo selection, and then maybe how AI can be applied in the future. So to date, we have been able to help a patient to determine their chance of success using this graph that maybe some of you would be familiar with, which shows the decline of fertility over time as a patient's age increases. But can we make this more specific to a patient when they attend if we include other variables? So we can look at the antral follicle count, the anti-Müllerian hormone levels, a patient's semen analysis, their BMI. We can also tailor it based on the current clinic that they're attending to see if the clinic's results also have an impact, and other lifestyle factors. Can these help to personalize a prediction of the patient's chance of success? And there are already AI tools out there. One of them is called Unify, which is a Canadian-based company, so a California-based company, where they're looking to specifically look at the patient and provide them with a more accurate assessment of their treatment chances. So this helps to personalize their success, allowing them to perhaps financially budget for the treatment because they have an understanding of how many cycles it may take to achieve their goal, help to balance their decision making on which treatments to perhaps adopt and what those success rates look like, supporting them all the way through that process. So in terms of moving towards the clinical aspects of our work, there are many areas where we can look to make improvements. So protocols are applied to a patient to try and generate the maximal number of oocytes without the potential for risk in relation to ovarian hyperstimulation. So can we tailor these? Can we make them more consistent to help guide our clinical teams in which protocol to use based on these patient demographic features that are linked to their success? We can also look at follicular scanning. As I mentioned before, this is done by ultrasound and therefore is a template to use artificial intelligence to infer perhaps more information than we could possibly get through a human assessment. Using these formats, can we then help to determine the actual timing of a procedure? An egg collection, and when it's done, may infer a difference in the amount of egg yield we may get, for example. Will an AI be able to support a clinician in knowing whether to continue medication, to timetable the appointment, or to cancel if it's likely that there is not going to be a yield of eggs? And once we've started using ultrasound, there's that potential to look at the endometrium, when to time a transfer and when's the best time for that embryo to go back into an endometrium. So this has the potential to individualize and maximize the protocols used for each patient to hopefully generate a better outcome, reducing the time perhaps they have to spend in the clinic to do this. Now moving on to eggs, sperm and embryos. Here you can see some images of an egg, sperm sample and a time-lapse image of an embryo that's in culture. And can we look at each of these parts of the process to try and maximize the selection of that embryo, as that is the key to a patient's success, by understanding more about each part of the process. So when we're looking at eggs, currently, when we assess eggs as embryologists, there's no real universal standard for quality. If the egg is mature, then we will inseminate it and hope for a fertilization event. But is there more that can be deciphered through the egg? We can often identify a poor quality egg, but we're not very able to differentiate a good quality egg. So could AI identify discrete markers, invisible to what we can see down a microscope, to help understand its capability of fertilization, or whether it's going to become a blastocyst, which is the precursor to an implanting embryo, or even an ongoing pregnancy? So we were very lucky to work with a company that had devised an AI tool looking specifically at eggs here at CARE. And this tool takes a 2D image from a microscope, a camera image from a microscope, uploads it to the cloud and assesses it immediately for competence and gives it a kind of range of prediction for either fertilization and blastulation. So we were a bit skeptical. Being scientists, we wanted to put this system through its paces. So we had 10 of our experienced embryologists pitch themselves against the Violet technology. We all looked at a single image of an egg, 300 images, and we assessed whether we thought that egg would either fertilize or result in a blastocyst. And as you can see by the score sheet there, we unfortunately didn't do as well as expected against Violet, with only one embryologist beating Violet for fertilization, and none of us were able to beat Violet for blastulation prediction. So the AI tool is obviously identifying something within that image that we are unable to identify, and the heat maps there show where the AI is focusing on in terms of the egg and its main areas of attention. And yes, the Violet system beat us in 12% of cases for fertilization and 18% for blastulation. So how do we use this technology in-house? We use it as a guiding tool for our patients, and a specific subgroup of patients utilize this, this technology. So we use it for patients who are attending for a social egg-free. So these are patients who have decided to pause their need for a family until later, and have decided to freeze their eggs at a younger age and then come back potentially in the future to use them when they're ready to start their family. But prior to this technology, the information they would receive at this point would be largely just egg number. So how many eggs had we frozen for them for the future? Using this new tool, we can give them far more information for decision making, for them to judge what to do as a next step. So this will identify the eggs, their egg quality. It will also identify from that group of eggs how many blastocysts might be achievable and how likely they are to get pregnant from that cohort of eggs in the future, using not just the AI, but demographic information on the patient, individual patient, and national statistics on outcome measures such as survival rates post-thawing of eggs and also live birth rates. So this helps inform the patient of their chance of success and helps to guide them on decision making as to whether to freeze some more eggs or to understand the potential if they were to return. So sperm, there's a lot of technology that's being created to look at sperm. Some of these include a home analysis kit that you can have an attachment for your phone where you produce a sample. You use the device that they send in the post to attach to your phone to get a video of your sperm sample. And this then can either be interpreted by an app on your phone, or it can be interpreted and sent to a clinic situation where an experienced embryologist or a CASA system like the one shown here, the lens hook, would interpret the sperm quality. Now, obviously, these are not, these are a lifestyle tool rather than a diagnostic tool, because currently they are not accredited for use for a diagnostic analysis. And then in terms of an alternative, not an AI device, but something of interest, which is the Zymo device. This is a microfluidic device that helps to separate motile sperm from the raw sample, but without the need for centrifugation, which has been linked to DNA fragmentation. So this helps to improve the quality of the sperm that we use to inseminate. And then taking this a step further, there's a company called IVF 2.0 that's developing a sperm selection assistant. Generally, when we're doing ICSI as practitioners now, we would look at a sperm and choose it based on its appearance and its motility. But SID has been trained to look at the motility assessments of the sperm and the morphology against a known outcome of blastulation or clinical outcome to determine from a cohort of sperm the highest ranking sperm. And here you can see them in green as they move around the screen. So this helps to direct the practitioner to the sperm of the highest quality within a large field of sperm. This is being presented at ESHRE in terms of a new technology. It's coming into commercial availability, and from early assessments, it looks like it can support a team with varying ranges of experience. It may not help enhance the already experienced practitioner in sperm selection, but will definitely provide consistency to those who have just started an ICSI procedure or are less experienced than their senior counterparts. So embryo selection, which is considered the most important part of a patient's treatment, determining the embryo with the best chance of implantation potential obviously will save time in terms of their fertility journey if we can identify that embryo from a cohort, because we generally produce more than one embryo when we proceed with IVF treatment. On average, you would get 6 to 8 embryos, and we want to know which one has the best chance of success. So there are many ways of doing this, and the AI technology out there is largely based on embryo selection currently. So you have technology that will look just at the image, so and determine the embryo's quality, or it will look at the static image and the time sequence events that determine an embryo's developmental capacity. So we have different formats that can either be a 2D static image or a time-lapse image, where we're taking images every 10 minutes, which is the example in the video here. That's determined from a time-lapse instrument. And these use both deep learning and machine learning to assess the embryo, the embryo's quality. What you want to determine as the output also determines which model you might want to use in-house, whether that would be ploidy status, so whether an embryo is normal and euploid or abnormal and aneuploid, whether you want to just predict whether that embryo will become a blastocyst in the future if you're doing an early transfer, or whether you want to see if it's got potential to be a live birth or a clinical pregnancy. So the value of what outcome you want obviously determines which tool you might want to utilize. So I wanted to go through our experience. We've been very fortunate. We've been doing time-lapse in our clinics since 2011, and this has generated a large data set for us to utilize to explore the potential of artificial intelligence. So we have skilled embryologists that to date have manually annotated every key event in the embryo's division. So that would be when the embryo divides to a 2-cell, then to a 4-cell, to an 8-cell, and then when it starts to blastulate. So many events have been annotated over many years by our skilled staff, and we've used a quality assurance program to ensure that these annotations are of gold standard. So we provided over 63,000 embryos to train this AI machine learning algorithm. These 63,000 embryos actually were 7 focal planes with an image taken every 10 minutes, so 5,000 embryos per images per embryo. So this equated to actually 500 million images that we use to train this current AI system, which is a massive amount of data. And we nearly broke Microsoft when we tried to transfer it to the company that were assisting us and had to do it in very small pieces of the pie. So what our outcome measure was, it needed to be at least as good as an experienced embryologist in identifying these key events. And it needed to work with an existing in-house statistical model that we use to identify live birth potential. And it needed to be also as predictive as our manual annotations when running this model. It had to be accurate and reproducible, and it had that measure of whether it would be streamlining our processes to save time. So manual annotation generally takes 2 minutes per embryo. Over 5 days, that could be as much as 20 minutes. So this had a big capacity to save us time in our laboratories, streamlining our actual procedures. So as I mentioned, we gave our data to a company called BJSS, who trained our AI model. It provided automated annotations. So instead of manually annotating, these annotations would be provided in seconds. We ran it with our existing algorithm, which we've published on, that is ranks embryos for potential for live birth outcome. We assessed within our laboratories. We did a parallel system to understand the time-saving elements, and we then ran the predictions on those manual and auto annotations to understand whether it was comparable to our manual annotation system that we would be replacing. So we did find that it was as good as our experienced embryologists, but there are alternatives out there. We're in a fortunate position to be able to identify and train and validate our own system, but other clinics may not have as much data to be able to perform this. So there are tools that are supplied by manufacturers. So Vitrolife have the embryoscopes and they provide guided annotation. And then we have other incubators with time lapse like the Jerry, which also have automated annotations, which guide the practitioner to a point in time to be validated by an experienced staff member. So we're not the only ones looking at the embryo. There are also commercially available systems. As I've mentioned, some of them look at ploidy. The first one there is a hatching blastocyst provided by IVF 2.0 and Alejandro Chavez Bandiola's group, who look at a static image of an embryo to identify ploidy status and rank the embryo according to its likelihood to be normal versus abnormal. The IDA score is a system provided by Vitrolife, which is one of the embryoscopes, which is a time-lapse incubator. They have created their technology on over 183,000 embryos, which have been gathered collectively from a diverse set of clinics. And these help support embryo evaluation within the teams. But what we would say, if you get the opportunity, now we've built our AI on our own data, so we understand the competence. We also understand the journey we undertook to achieve that AI. And as I say, we have great transparency on the data that was used. If you get the opportunity, you should ask these commercial entities whether you can retrain on your own data. And if that's not possible, obviously to run an in-house validation to confirm that the results that they are achieving marry up with your own systems. So looking to the future, and where we might be going with AI. We've looked at some of the possibilities that are out there currently, whether they're still in evaluation stages or whether they're a commercial entity. But there's also movement within our field to look at automation, whether that's trying to automate ICSI and provide some consistency that may exist within practitioner variation. And then also there's been talk of a laboratory on a chip where all of these technologies can be combined. So whether that be metabolomics or non-invasive PGTA and time-lapse, all of this will generate many data points that will need to be assessed. And AI may only be the facility that will offer a readable variable once all of these treatment technologies are combined. So in summary, there is this prolific explosion of AI within our field. We cannot go to a conference without seeing many different presentations and posters on AI within fertility care. It is generally designed as a support decision-making tool, and it's there to assist and not replace experienced clinical staff and provide some consistency when we're interpreting complex data sets. It has the potential to save time, being more reproducible and accurate perhaps than a manual variable, which is open to subjectivity. And with personalised patient care, we would hope to improve the potential outcomes for our patients who are attending our fertility clinics. But it comes with a cautionary note. There's been a lot of talk about black box technologies. Understanding how it comes to that result is important as a scientist to understand that journey and understand what that may mean, and that may not be possible in some of these technologies. And in our field at the moment, there's a little or lack of regulation. We are governed by the HFEA, and at the moment they are playing potentially catch up with some of these circumstances as this technology is being proliferated by commercial entities. And we do need, again, that RCT to confirm and validate validate that these technologies are working. And if that's not possible, then obviously in-house validation is advised. So, so yes, I believe that in summary, AI will be present within our field in the future. How these tools are utilised obviously needs to be regulated and understood, and we need to be clear on the advantages and the benefits that it's offering to our patients. So thank you very much.
Thank you very much, Rachel, for a walk through fertility and AI. Fascinating, thank you. There are a number of questions that have gone into the chat box, but we are running a little bit late on time. There are two questions which are directed towards the panel. So I would ask Dr. Bahadar Bhatti from Birmingham. Could you please ask your question that you've put in the chat for the panel?
Thank you, Stephen. Can you hear me okay?
Yes, thank you.
Perfect. My questions really relate to making sure we've got a governance framework when implementing AI. So the first question is, to what extent do the clinical users recognise the risks in using AI decision making, and what steps are in place to mitigate them?
Who'd like to kick off?
I'll have a go, shall I? Users, there's no evidence that there is a risk, particularly with something like speeding up MR. I don't think there's been sufficient phase 4 type of analysis on the data, or when things were missed, or when additional lesions are identified, so false positives, etc. So I don't think they appreciate the risk. And again, it depends on the AI tool. You know, the orientation of the ultrasound is, it essentially just, it flashes green when they've got the right orientation, and they take the image. So that carries very low risks. But I would think that there needs to be the phase 4. We've done, in some senses, we've done enough phase 3 like experiments with some tools that it can only really be, that question can only really be answered with market surveillance, as they call it, as phase 4 types of data collections.
Thank you. What I'm really getting at there is cognitive bias, in the sense that how does it affect their decision making, and to some extent you really have to think about where you put the AI in that clinical workflow.
So there's a recent paper that came out of Sweden that looked at the influence of a breast screening tool on observers and on readers, and the least experienced readers are the ones that are most affected in as much that their kappa, their agreement with the AI tool, goes up. Yeah. Now, the more experienced readers, the impact was almost negligible with the AI tool. Having said that, the dual reading system has an influence, in as much if the first reader is inexperienced compared to the second reader tends to ignore the first reader's opinion. Yeah. And vice versa. If the first reader is experienced and the second reader is inexperienced, they tend to agree with the first reader because they're more experienced. They essentially, so it happens with humans as well as AI human interaction. So I think that's where I'm coming from. Will it diskill people in the future? I think only the future can tell that.
Thanks, Roger. Rachel, would you like to chime in?
Yeah, I'm happy to have a go. So when we're talking about de-skilling, we would argue that our roles change over time. Some of these mundane tasks maybe are something that we don't need to have as part of our repertoire. We've moved from making media back in the day to now buying commercial media, and I don't think any embryologists would argue that they've missed making their media, filtering, pH testing it. So there are some parts of our process that I think AI can help to replace, and the embryologists will use those tools to make a decision, not be overrode and the embryologist will ultimately have the final say, and these decision tools currently are just there to provide further information for that decision to be made. So that's where we are currently in our field. But yes, with the movement of technology, I can understand that in the future that may not always be the case, but tools at the moment are just there to enhance the experienced practitioner's decision and not to replace it.
Yeah,
Thank you, Rachel. Ashley, do you have a quick comment? We are quickly running out of time here, so 30 seconds, please.
Yeah, I mean, I think I'd largely agree with what Roger and Rachel have said, really. And I think there's an interesting study recently for histology showing that on the skill side of things, AI can actually be used to help enhance those skills. Where when training people to read slides, if you have an AI assistant there, then they can actually learn faster. And I think that is a kind of an interesting use case, really. That's kind of maybe a counterpoint to that.
Lovely. Thank you very much. Just leaves me to say thank you to the speakers, Roger, Ashley and Rachel. You can go back out into the sun. Thank you. Thank you all very much for your input and very stimulating series of talks. Thank Kirsty for the organisation together with Brendan, and thank you all very much. Bye-bye.